Be the video on blood vessel structure and atherosclerosis, and probably capillary exchange too. And then we'll do a separate video on blood pressure regulation. All right, so if we take a look at this diagram right here, we can see the difference between an artery and vein as far as their structure is concerned. This micrograph shows how thick the wall of an artery is, whereas the wall of a vein is very much thinner. Most arteries and veins are found right next to one another as the blood is leaving the heart, going away from the heart and coming back towards the heart. You'll also notice the lumen, or the opening inside of a vein, is much larger than the lumen of an artery. That difference has a lot to do with blood pressure regulation and ensuring that blood is always flowing away from the heart and coming back to the heart. So if we take a look at the layers of the blood vessels, let's go from the outside in. So let's start here with the tunica externa. Sometimes it's the tunica adventitia. A lot of times things are exactly the same thing, and it depends on where you are, is the terminology that we use, but I like to use tunica externa. You can see that this is essentially similar to the serosa that we have in most of our organs in our body, and there will be a lot of collagen that makes up the tunica externa, and then you'll also see that there is a vasovasorum. And one of the questions that is asked in your study guide is, how do the cells comprising the different blood vessels receive oxygen and nutrients? And it's through the blood vessels that we find in the wall of a blood vessel. And as long as the blood vessel is rather large, the vasovasorum is quite abundant and quite noticeable. And then when we get down to the capillary layer, then it's just going to be simple diffusion that will allow the nutrients and the wastes to cross back and forth through the endothelial walls that form the capillary. It's really interesting in a cadaver to see in the aorta, for example, in the pulmonary trunk, some very large vessels, and to see the vasculature that's running through that tunica externa of those large vessels. Then as we go in, we have an external elastic membrane found only in the arteries. That's going to be between the tunica externa and the tunica media. The tunica media is the smooth muscle layer. You'll notice the thickness of the smooth muscle is greater in an artery than there is in a vein, and we'll explain in blood pressure regulation the significance of that. This thick smooth muscle layer is also what this pink and purple layer is right here. This outer, more red color is the tunica externa, and then all this is tunica media. You can see there's only one or two cell layer thick here of smooth muscle in the corresponding vein. And then we have another internal elastic membrane, and then our subendothelial layer, which is analogous to the basement membrane that we find in this endothelium here. And then we have the endothelium in each blood vessel. You can see the subendothelial layer is slightly thicker in a vein than it is in the artery. And then the endothelium is pretty much the same between an artery and a vein. Because of the thickness of the wall, again, the lumen of an artery is much smaller than the lumen of a vein, and coming off of the endothelium of the vein, you will see a valve. And this valve the flaps of the valve will point towards the heart, and so they will open distally from the heart and close proximally to the heart as blood is flowing back towards the heart. This discourages the backflow and pooling of blood, as there's so very little pressure generated within a vein that gravity would have a large effect on the blood flow here in the vein. Also, valves are typically found in the extremities, and we don't usually find valves within the blood vessels within the trunk. Then if we take a look at the structure of the artery as we go out, these would be arterioles here, and each of these walls would be thinner until finally we get down to the capillary, where we have lost the tunica externa and the tunica media, and all we're left with is this tunica intima, which is, or tunica interna, either one, which is simply the endothelial cells and their accompanying basement membrane. Then as we leave the capillary bed and we build back to go back into a vein, we will increase the thickness of the walls and put back the missing walls until we have a vein again. Okay, so let's go to the next as we look at the various types of valves. Oh, let's talk really quickly about incompetent valves, because I don't think that we really discussed that in your study guide. No, we didn't. Some of you may have heard of varicose veins. Elderly individuals, often if you look at the blood vessels in their legs, that the vessels all just kind of look like an old river that's just meandering through their skin, and that's due to valves that are not working anymore. Too much pressure, too much back pressure on those valves will allow the backflow of blood, and then for it to pool in the veins and then cause them to be tortuous or twisty, and that's what a varicose vein is. And so when the valves within a vein don't work anymore, then typically you're going to get varicose veins. Also, because of the pressure, either because you're pregnant or because you're constipated, you can get varicose veins in the anus, and that's what we refer to as hemorrhoids. So that's what happens when your valves don't work. Okay, so here is just a description of the blood vessel wall or layers. This is most important. Okay, is we want good simple squamous epithelium, slippery, very tightly bound together with tight junctions, and no opportunity for the platelets to cling, as we saw in hemostasis. Oh, this is very important. I'm so glad that I saw this. Forgot to tell you that the tunica media, primarily of the arteries, is innervated by vasomotor fibers coming from the vasomotor center of the medulla, which we will talk about in the heart regulation of cardiac output. And so smooth muscle is innervated by sympathetic division. There is no parasympathetic innervation of vascular smooth muscle. It's only under control by sympathetic. So when the alpha adrenergic receptors, when norepinephrine and epinephrine bind to the alpha adrenergic receptors on vascular smooth muscle, then we get constriction. So the smooth muscle will contract and we'll get vasoconstriction. Conversely, just to tell you now, and I'm sure I'll reiterate this later, but the smooth muscle in the muscles, on the other hand, have beta adrenergic receptors, and in the presence of epinephrine and norepinephrine are going to vasodilate. And so this is the only example where we have vasodilation of a blood vessel under nervous control. Typically, if a blood vessel dilates, it's because there's nothing telling it to constrict. And so that's why with in this particular case, this vasomotor fibers of the sympathetic division, we tell it to constrict, but we don't have to tell it to relax and dilate by parasympathetic. So no parasympathetic innervation of vascular smooth muscle. Okay, so there's the tunica externa, and here's vasoconstriction and what vasodilation is. And here are the blood vessels. So if we take a look at, describe the various types of blood vessels that a red blood cell flows through as it travels from the heart to the body and back to the heart. Here is the blood vessels that leave the heart are the elastic arteries. Elastic arteries don't mean that they have a lot of elastic in their walls, but they have elastic properties. The blood pressure leaving the heart is very strong, and so in order to prevent any damage to the walls of the vessels, as the blood pushes out on the walls of elastic arteries, they expand, and then they recoil. We also refer to these as conducting arteries because they are conducting blood away from the heart and to the major organs of the body. Typically, wherever you can feel a pulse, that's going to be a conducting or an elastic artery. Then we get to the muscular arteries, and these are muscular because they have a strong or a thick smooth muscle layer, and they are distributing to the basic tissues, or the tissues of the body. These are typically the ones that we name in lab, and they are muscular because of that thick smooth muscle layer that they have. Helps maintain the flow of blood by maintaining adequate blood pressure. And so this is where we see a lot of the vasoconstriction, vasodilation going on that we mentioned before with sympathetic stimulation. Then we go down to the arterioles. We call these arterioles resistance vessels. They are almost always constricted. Force from the heart is very low by this time, and in order for blood to flow, it must always flow from areas of high pressure to low pressure. So we need to make sure that the pressure is adequate in the arterioles as it is about to encounter all the tissues of the body through the capillaries. So vasomotor tone, very, very important in the arterioles. They are almost always constricted. Then we go to a terminal arteriole, which is going to lead to a specific capillary bed. And then we have our capillary beds. And I'll talk about the sphincters in just a minute. Then once the blood has been in a capillary bed, then the capillaries are going to unite. So we have blood vessels that are dividing and dividing and dividing as they move away from the heart, and then they are coming together as they come back to the heart. So we have branches, and then tributaries as they're coming back. And so the capillaries are going to come together and form the postcapillary venules. One thing you'll want to remember about a postcapillary venule is that this is where inflammation or the effect of histamine during the inflammatory response is going to occur. When blood vessels get very permeable in the presence of histamine, it will be the postcapillary venules that will leak, and where the white blood cells, the diapedesis that will go on, will come out through the postcapillary venules. Then those postcapillary venules are going to come together and make small veins. We call these small veins, and then large veins. You'll notice in this picture the presence of valves in these small veins. We call these capacitance vessels, both with the small and large. Because of their large lumen and their low pressure, blood collects in these, and at any one time there's 65% of our total amount of blood, the 5.25 liters that we typically have in our body, 65% of that will be found in the capacitance vessels. And it's really important that we understand there's a lot of blood just slowly making its way back to the heart when we talk about increasing cardiac output, when we go back to the heart notes, and how venous return increases end diastolic volume, and then end diastolic volume increase will allow preload to happen, and then we increase our stroke volume, and we decrease our end systolic volume by that preload increasing the contractility or the force of contraction of the ventricles, and we increase our ejection fraction. So as we get back to the large veins, then we have returned back to the heart. So we go elastic arteries, muscular arteries, arterioles, capillaries, postcapillary venules, or venules, small veins, large veins, and then back to the heart. And so we have a closed circulatory system because blood is always, under normal conditions, when we're in homeostasis, blood is always contained within a blood vessel. Only exception that we have in the spleen. We have some sinuses in the spleen, a little bit in the liver, where the blood isn't necessarily in a very narrow and tight blood vessel. Okay, so let's go to the capillaries. I want to do the sphincters, where the sphincters come in. Oh, okay. So on the back of the study guide, well, on the second page of the study guide, it says describe the structure of the function and purpose of precapillary sphincters. So there you can see them in this picture. They are going to come from an arteriole as we go into a tissue bed or a capillary bed, and they are going to close both under sympathetic and chemical stimulation. So nervous and chemical stimulation will close and open these sphincters based on where the blood needs to be at any one time. We only have, like I said, 5.25 liters of blood to deal with, and so if some areas need more blood at a particular time than another, we have to move that blood to those areas that need more blood and keep it away from the areas that are not as important. And so by shutting down these precapillary sphincters, we're able to send the blood either through an anastomosis. So here you can see an artery and a vein are connected together with no capillary bed in between, and that's an anastomosis. It's a hurry up way of getting blood back through the arterial and back into the venous circulation, and then this thoroughfare channel. So if we shut down all these precapillary sphincters, then the blood has no other place to go but through this thoroughfare channel, bypassing the capillary bed and then back into venous circulation. So an example of when I would want to shunt my blood, sympathetic stimulation, for example, because I'm running away from a Rottweiler. And my muscles and my heart and my brain and my lungs need lots and lots of blood going to them and not so much blood going to my digestive system. And so the capillary beds of the digestive system will have their precapillary sphincters shut, and the blood will then bypass them and then go back into circulation and end up out in the muscles, wherever we need them to help us run away from the Rottweiler. Then when the Rottweiler's gone and everything is safe, we have to digest the lunch that we just ate. And so we're going to bypass the capillary beds in the muscles, and we're going to take the blood back to our digestive system. And then when we get into the kidneys, then we'll talk a lot about shunting blood and what we do to help regulate blood volume or fluid volumes when our kidneys are involved. Okay, then we just have these explanations of, so here you can read through that if you pull up the PowerPoint, and then muscular and the arterioles, and now we get into the capillary structure. So we have capillaries, again, are only endothelial, so simple squamous epithelium and the accompanying basement membrane. And that is all that they're made of. So the wall is very, very thin, and squamous cells are also very, very thin themselves, and so it's very easy for diffusion to occur. So material can diffuse from the bloodstream into the interstitial fluid outside of the blood vessel and into the tissue beds, and conversely, the material, secretions, waste products, whatever needs to leave the interstitial fluid in the tissues and go back into the bloodstream will very easily be able to diffuse back into the bloodstream. So that's what's appropriate for exchange there in the capillaries, is that their structure. And I was just going to say something else about them. And they're very, very narrow as well. And so you can see that red blood cell right there is how big it is, and the capillary just barely goes around it. So you can see they're very, very tiny, and by being very tiny, we can have a whole lot of them, and so that we can increase the surface area for exchange of gases and nutrients. Okay, so we've got 3 different kinds of capillaries. So the top one is a continuous capillary. Notice it says it's the least permeable and the most common. This is where we're going to have no openings in the wall of the capillary. This is very, very important. We have in the brain, not only your brain is so picky about what can get into cerebrospinal fluid that not only does it have continuous capillaries, but it has a blood-brain barrier as well. I'm sure you probably learned that in 264 with the astrocytes. The brain does not want a lot of the stuff that's traveling through your bloodstream to get into its cerebrospinal fluid. And then skin, muscle, most places, like it says, is the most common capillary that you have. So you can see the walls, they're tightly held together. They do have that basement membrane that goes around them, lots of tight junctions. Then we go to the fenestrated capillary. For those of you that speak French or Spanish, this is how I always help my students know fenêtre in French means window, and then ventana in Spanish is also window. So my students always know fenestrated capillaries have windows in them. And these pores, or these large fenestrations, notice increase their permeability. Obviously, more things can get through if there's openings. We still have an intact basement membrane that the material would have to diffuse through, but we want to hurry up to get things in and out. Because basically we can't slow down the blood and we want things to move, or they're too large to be able to diffuse rapidly through the cell membranes of the endothelial cells. So where do we have that? Areas of active absorption or filtration. So we have them in the kidneys. The glomeruli of the kidneys are fenestrated capillaries. The capillaries that we have within the villi of the small intestine are also fenestrated capillaries. Also, the blood vessels that we have in endocrine glands are also fenestrated, because those hormones that are coming through are too large and too polar, and they just can't slip through the cell membrane and then get into or out of the bloodstream. And then the last ones that we have are the sinusoid capillaries. And sinusoid capillaries, notice we have an incomplete basement membrane, and we have large openings, large spaces, intercellular clefts. And these are the most permeable, and they are going to be in areas where we need cells to be able to pass back and forth, red blood cells, white blood cells, platelets. And so we need large openings there. So the liver, as we're processing the nutrients and the blood in the liver, and the bone marrow, the red blood cells need, as they get made, can now enter the bloodstream and go throughout the body from the bone marrow. And then the spleen is, of course, where we're going to take out the dead, they're always dead, the raggedy old red blood cells that need to be recycled.